Okay, so good morning, everybody. In our last class, we discussed the chemical level or chemistry of life. So today we will talk about the cellular level. You know that cellular level deals with the cells and the organelles, right? So we will basically talk about the cells and how the cells work and the parts of a typical cell. Okay, you know, human body consists of hundreds of billions of cells, not thousand, not million, billions and hundreds of billions of cells. So now we can just think how small, tiny a cell is, and you need the microscope to study the cell. Without microscope, you won't be able to see. So, why cell is called the structural and functional unit of the living body? A cell can individually survive. That's the smallest component of the body that can individually survive. That's why it is called the structural and functional unit. Now, let me give you an example. How you know that cell is the smallest structure that can survive individually. You know that bacteria, some microorganisms, right? Amoeba, those are single cell organisms. They can survive. If I take cells from your body, like a skin, and put in a petri dish in solution, give oxygen and nutrition there, the cell will survive. Individual cells will survive there. We can keep them alive. So, you know that cell is the smallest component of the body that can individually survive. Now, if I break the cell membrane, then what will happen? It will die. If I break the cell membrane, it will die. So, that's the smallest structure. So, first we will see the definition of a cell and the parts of a typical cell. Then we'll talk about the cell membrane. We'll see the structure of the cell membrane and the function of the cell membrane. Then we'll talk about the proteins present in the cell membrane. In the cell membrane, there are two types of proteins and they perform some very important functions. So we'll see what are those two types of proteins in the cell membrane and what are their important functions. Then we'll talk about the junctions. What are the junctions? Junctions are the structures that attach the or connect the cells, one cell to another. So we'll see different cellular junctions and their structures. Then we'll talk about the transport across the cell membrane. Living cell membrane is a semi-permeable membrane. That means it allows certain chemicals pass through it, but not everything. That's why it is called semi-permeable, partially permeable. Make sense? So we'll see how transport occurs across the cell membrane, through the cell membrane. Then we'll see the response of a living cell in three different types of solution. if we put a living cell in isotonic, hypotonic and hypertonic solution, how that cell will respond. Then so those are the things. So first, definition, I have already mentioned. Cell is the structural and functional unit of the living body. Human body has 50 to 100 trillion cells. So trillions, 50 to 100 trillion. And over 200 different types of cells are present in the human body. How they are different? They are different in shape, size, color, function. We will see few examples how different cells are different. But just know that there are over 200 different types of cells present in the human body. But one thing is common for all the cells, the chemical structure. All cells of your body are composed of CH, N and O, carbon, hydrogen, nitrogen and oxygen. You must remember from last lecture that 4 elements form 96% of body mass. Remember, these are those 4 elements. So our body is mainly formed by the cells. So these are the 4 elements or chemicals. Okay, now how the cells could be different from each other. Here are a few examples. You see on the top left, those 2 cells are fibroblasts surrounded by fibers. These cells, fibroblasts, are connective tissue, most common connective tissue cells, and they produce fibers. You know, in connective tissue you must remember in many connective tissues there are fibers, right? Collagen fibers, elastic fibers, you must remember. So synthesis of fibers. That's why they are called fibroblasts. In the top right, you see those 2 red colored round flattened cells. Those are the red blood cells or erythrocytes. And red blood cells are different than other cells. How? They are round but flat, not spherical. No nucleus is inside and the color is red. And the function is also highly specific function. Red blood cells transport oxygen. So you see, they are different in color, shape, right? And function. And then if you see next one, those 5 cells are attached side by side to each other, and those cells are epithelial cells. And this type of epithelial cells are called cuboidal epithelial cells because of their shape. You see, cubical shape, cube. Then if you see those 2 red colored long, the red colored cells below the cuboidal epithelial cells, you see in the left side that long one. Inside that there are multiple nuclei. You see purple color. That is a skeletal muscle cell. Skeletal muscle cell is very long. You don't need microscope to see. Several centimeter, could be several centimeter long. And in the right side, those few cells are another type of muscle cells. Those are smooth muscle cells, and they are much smaller and they have single nucleus. So smooth muscle cells have single nucleus, but skeletal muscle cell has multiple nuclei, and you see even different types of muscle cells are different. Now in the bottom left you see that is a fat cell or adipose tissue cell. The nucleus is located not centrally but attached to the membrane, and most part of this cell is empty inside. Only few fat droplets are present. Function: storage of energy. You know that fat stores energy, so that is the function of fat cell. Uh, then in the right, bottom right, that is a macrophage. Macrophage has multiple processes. You see extensions from the cell body, and with the help of those processes, macrophage can engulf or do phagocytosis, engulf the microorganisms or dead cells of your body. So they can eat up other cells. Right top, that is a neuron or nerve cell. The neuron has multiple processes. You see, from the cell body, short processes, and then one long process is there. So, multiple short processes and a single long process. That is a typical neuron. Function: electrical signal transmission. Electrical signal transmission is the function of nerve or neuron. So you see, look different, function is different. The bottom one, the last one here, is a sperm, male sex cell. And you know that the sperm fertilizes the egg or ovum. You see, the sperm looks like a baby fish. It's an interesting structure. It has a head, mid-piece or body, and a tail, long tail, right? So it can swim in the fluid. So just few examples how different cells are different in terms of their shape, size, color, function. We have more than 200 different types of cells in the body. Okay, so now we'll see the parts of a living cell. If you see a typical cell has a membrane that is called the plasma membrane or cell membrane. Okay, that gives the boundary of the cell, and then you have a nucleus usually located in the center, and in between you have the cytoplasm. Okay, so in between the plasma membrane and the nucleus you have the cytoplasm. Now, cytoplasm has two components in it. What are those two components in cytoplasm? In cytoplasm, the liquid part is called cytosol. That's the liquid part, and you have organelles. So, you have two parts in cytoplasm. The liquid is the cytosol, and organelles are the structures like mitochondria, Golgi apparatus, endoplasmic reticulum, lysosomes, centrosomes, ribosomes. All those are the organelles. Okay, so you see, these are the parts of a typical cell. That spelling, it's I made a mistake here, or the N-E-L-L-E-S. Okay, so now the plasma membrane as I have mentioned, gives the boundary. It separates inside the cell from outside, and it also gives the shape of the cell. So two important functions of plasma membrane, just remember. One is it gives the boundary that separates outside and inside the cell, and number two is what? It gives the shape of the cell. And the plasma membrane is a semi-permeable membrane that I have mentioned before. Now, nucleus. Nucleus is called the control center. Control center. Why? Because inside the nucleus you have the most important component of a cell. Those are called DNA. DNA are located inside the nucleus, and DNA controls the functions of the cells. So that's why nucleus is called the DNA. We know that DNA also transmits the hereditary information, right? From the parents, the information, the characteristics pass to the offspring. So that is also the function of DNA. So DNA regulates the cell function, how the cell will behave, what will be the functions of the cell. Not only that, transmits the information to the offspring. Anyway, so now different organelles. We'll talk about few important organelles. I believe you have learned these things a lot in your high school, right? Yeah. In biology. So, but just briefly. So, important organelles are mitochondria, Golgi apparatus, endoplasmic reticulum, ribosomes. So, let's first see the mitochondria. Those orange colored structures, organelles, are mitochondria, and mitochondria are known as the powerhouses of the cell. Why? Because inside the mitochondria ATP, most of the ATPs are produced inside the mitochondria. So that's why you know that ATP provides energy, right? You have learned that in last class. So that's why mitochondria are known as the powerhouses of the cell. Then around the nucleus, in this picture, you see those blue tube-like structures. Those are endoplasmic reticulum, or ER. Now, they are located around the nucleus, and they look like many tubes are connected to each other. Okay, so many tube-like structures are connected to each other. That's the endoplasmic reticulum. Now, endoplasmic reticulum is two types: rough ER, endoplasmic reticulum, and smooth ER. One part is called rough because if you see the surface of rough ER, you'll see many ribosomes, like tiny dots, are attached on the surface. So these are the ribosomes. These are the ribosomes attached to the on the surface, and that's why the surface is rough and it is called rough ER. Another part doesn't have ribosome attached, so smooth surfaced ER. Okay, so the rough ER and the smooth ER, they perform two different functions. The endoplasmic reticulum is responsible for protein modification of proteins. Then what happens actually, the ribosomes on the surface of rafia, these ribosomes manufacture proteins. So protein manufacturing or production is the function of the ribosomes. Then after the proteins are produced by the ribosomes, then the proteins enter into rough ear, because ribosomes are just attached on the surface. So from there the protein molecules enter into the ear, and inside the rough ear that protein is modified. What is that? You know that protein has amino acids, right? Amino acids get connected to each other to form the protein chain. So rough ear, sometimes it cuts the protein chain to make it in right size, the length. So rough ear modifies the protein by cutting and folding, those kind of things. So rough ear, let me write down here. So rough ear function is protein modification. Cutting the protein chain and folding. Okay, so that is the processing of the proteins. Then rough ear sends the protein to another organelle, that's the Golgi apparatus. So from the rough ear, protein goes to the Golgi apparatus, and Golgi apparatus does what? Packaging of protein. Okay. So you see, interestingly, these functions are like, what happens in a factory. In a factory, if you go to a factory, what happens? The production, right? Manufacturing first. So who does the manufacturing of protein here? No, manufacturing. Oh, the ribosomes. Ribosomes. Ribosomes manufacture the proteins. Okay? Make sense? That's the production. Then ribosomes send the protein to rough ear, right? Endoplasmic reticulum. Where what happens? Further processing, like folding, cutting, right? Those things happen there to make them in right size, and then fold them. And then from rough ear, protein molecules are sent to the Golgi apparatus, where the last thing happens. What is that? Packaging of protein. Make sense? So it's like a factory. Production, then folding, and then packaging. Make sense? So three structures perform those three functions. Now smooth ear. Smooth ear has different functions. Number one, detoxification, a very important function. Inside the cell, toxic chemicals are also produced because metabolism occurs, right? Metabolism produces some toxic chemicals. So always toxic chemicals are being produced inside your body, inside the cells. So those toxic chemicals are being detoxified, neutralized in the smooth ear. Number one, very important function. Number two, also when you take medicine, that all medicines have some chemicals that can cause harm. So those chemicals are also detoxified in a smooth ear. Make sense? So detoxification, number one. Number two, lipid synthesis. Synthesis of lipids. Number three, synthesis of certain hormones. Okay, so just know those couple of functions of smooth ear. Then what the Golgi apparatus does, if you see the Golgi apparatus, the green structures here in this picture, it looks like a number of flattened sacs. Number of flattened sacs are attached to pile to each other on each other like this. So flattened sacs are piled together, and both ends are expanded or wider. And what happens, you know that from rough ear, proteins are sent to here. So protein molecules enter into the Golgi apparatus, and these protein molecules move to the ends of these flattened sacs. So go to the both ends like this, and then what happens? The ends get separated, detached, and form round membranous structures, and these are called vesicles. Okay, or also known as lysosome. Okay, same thing. So lysosome is in the picture. The round membranous structures, those are filled with protein molecules. So that protein could be hormone or something else. So that's how the Golgi apparatus produce the lysosome. So all lysosomes are round membranous structures. Those are filled with different types of proteins. Within the cell, you know that sometimes hormones are produced, some cells produce hormone. So if that's the function of that cell, production of hormone and secretion of hormone, then the vesicles will be filled by that particular type of hormone. So yeah, cell membrane or plasma membrane. If you see the structure of the cell membrane under the microscope, it is a phospholipid bilayer. So if I just touch a part of cell membrane and see under the microscope how it looks like, it is a bilayer of phospholipid. Bi means two, so two layers of phospholipid. These are the phospholipid molecules, okay? So this is one layer of phospholipid molecules, and another is like this. Okay, so two layers of phospholipid in which proteins are embedded or inserted. So this is a protein, phospholipid bilayer. This is another protein. So now you see that this is the cell membrane, structure of a cell membrane under very powerful microscope, electron microscope. So these are the proteins, and this is one layer of phospholipid. This is another layer of phospholipid. That's why we say that the cell membrane is a phospholipid bilayer in which the proteins are inserted or embedded. Now, if you see one phospholipid unit or molecule, it looks like this. This is called the head part of phospholipid, and these are the tail. So a phospholipid molecule has two parts, head part and a tail part. Head part is also called polar end and tail part is called non-polar end. Make sense? So, two parts in the phospholipid molecule. Now, polar end is hydrophilic. Hydro means what? You know that. Hydro, water, right? Philia means liking, attraction. So, this head part loves water, likes water. That's why this part is called hydrophilic. Tail part is hydrophobic. Phobia is fear or scared, disliking. So tail part doesn't like, this part doesn't like water. So now let's see here. So if you see this picture, this part is what? Hydrophilic or hydrophobic? This entire part? Hydrophobic, right? Because tail part is hydrophobic. And this entire middle portion is hydrophobic part. Is it clear to everybody? This is hydrophobic, right? Because tail part is hydrophobic part, this part. Only this part is hydrophilic, that means like water. That's why we say that cell membrane, most part of the cell membrane doesn't allow the water molecules pass through it. Make sense? Because it will repel the water. Because most part, you see here, is hydrophobic part. Is it clear? So water molecule cannot easily pass through it. If it loves, then water molecule can pass through it, right? If it doesn't like, then water molecule will not be able to pass through. So that's why water molecules cannot pass through this phospholipid bilayer. But we know that cell needs water, right? Water must get in. So that's why you have these protein structures. There are channels and water molecule and other molecules can pass through, water soluble molecules can pass through this. Okay. So that's the phospholipid part. Now proteins. There are two types of proteins in the cell membrane. Those proteins are inserted in phospholipid. You see here two proteins. Both are inserted in phospholipid. This one is completely inserted all through. This one is partially inserted, but both are inserted in phospholipid, and these are called integral proteins. Integral proteins. Now there are proteins, those are attached to the integral proteins, but those are not inserted in phospholipid. These are just attached to the outer or inner part of the integral protein. These are called peripheral proteins. They are attached to the integral protein but not inserted in phospholipid bilayer. So those are two types of proteins, integral and peripheral. Both are very important because they perform some important functions. The structure of the cell membrane is explained by a model that is called fluid mosaic model. Why we say that? If you see the cell membrane under very powerful electron microscope, what it looks like is a beautiful, white floor. Beautiful white floor, in which white floor is the phospholipid part, lipid, this part. If you look from the top, just think that you are looking from the top. This is the white floor, and in which beautiful marbles are inserted. These are the proteins. So if you look from the top, phospholipid, white flour, and the protein structures look like the marbles or stones inserted in. Make sense? And why it is called fluid? Because the protein structures are changing their shape too. So it looks like the floor is moving. That's why it is called fluid mosaic model. So two types of proteins, integral and peripheral. Just remember the important functions of these two types of proteins. As I have mentioned, both perform important functions. Integral proteins help in transport because they have channels and some of them work as carriers. Integral protein also act as a receptor. So here these are the important functions of integral protein. Transport, number one, acting as receptors. Some integral proteins work as receptors. I will explain that to you. Just know that. And some integral proteins form channels. And another function is just add some integral proteins work as enzymes. So those are four important functions: transport, acting as receptors, forming channels, and acting as enzyme. Transport. How some integral proteins help in transport, you see here. Two integral proteins are shown, and in the left side one there is a channel through it like this, and this channel always remains open. So the smaller molecules, those are smaller than the size of the channel, easily can get in or out because that channel always remains open. Another integral protein you see here, this one has a channel too, but this channel is a gated channel. That means it can get closed and open. So one is always open, right? So smaller molecules can easily get in and out through it. Another type of channels that are present in integral proteins, some integral proteins, those are gated channels, so they can get open and closed. So to open them, you need ATP. So in this case, ATP is required, energy is needed to open the gate and close the gate. So two types of channels can be present in the integral protein. Acting as receptors. Some integral proteins work as receptors. What is that? Let me show you here. You see, this is one integral protein. This is another integral protein. Let me draw another one here. This is another integral protein. Okay. Now, this is outside of the cell. This is inside the cell. The outer end of some integral proteins, you have the binding sites. For example, this one has a binding site and the shape of the binding site is square. This one has a binding site, shape is triangular. This one has a binding site, shape is round, circular. Now, if a hormone wants to work on this cell, the hormone comes from outside, we know that, and if that hormone molecule, the shape of the hormone molecule is round, so this is the hormone, that is called the signal. So signal molecules or hormone molecules will come, and since these molecules are round, they will perfectly fit here. If it goes there, no, because the shape is different. If it goes here, no. Okay. So this is the receptor protein for this hormone molecule. Make sense? Signal. And when that binding will be perfect, then the signal will go to the inside the cell and intercellular machineries will start to work. That will stimulate the DNA and protein synthesis. So just know that what is important, the receptor signal binding. This is very important. The binding, perfect binding is essential for the signal to work. So this is the binding. Signal receptor signal binding. Now if you take a medicine, sometimes we take medicine, right? If you take a medicine, that medicine also works on the cell. So if the medicine molecules are like this, we'll go here, now, go here, now, but here the molecule will bind perfectly and the signal will go inside the cell. Make sense? So that medicine will work on the cell. If the cell membrane doesn't have any of those shapes that hormone or medicine is going there, then the cell will not be stimulated, will not work. So that's how the integral proteins work as receptors. And it is like key lock system. You have the perfect key of your car, right? For your car, if you take another person's car's key, will not start the engine. To start the engine of your car, you need that perfect key, right? That will fit and then you will be able to turn on. So that fit must be perfect to work on the cells. Is it clear? So that is important.
Is there an infinite number of protein binding sites, like the shape and the different? Is it just like does it just coincide? Is it just like an infinite number, like they haven't discovered like every single type of binding site, or are they all discovered?
Not all discovered. Particularly, we don't know many of the receptor binding sites in the nervous system, particularly. Okay, but other structures we know more or less. Yeah, yeah. But this is one important research that is always going on. The drug manufacturers, they must know if they want to produce a medicine that should work on the liver or specifically on the kidney, they first need to study the receptors present in the kidney or liver, right? What's the shape of those receptors? Then they will make the molecules like that. So we'll go there. We don't want all medicines work on all the tissues or structures of the body. That will be dangerous, right? So we must be very careful. And a lot of these molecular research, are done before they make the medicines.
So we want anything to be very specific, basically.
As more specific you want, yes.
The more specific it is, the more it's going to direct to the
that target organ, yes. Some integral proteins work as enzyme, and you see here they have just shown two integral proteins, and they're converting the molecules, one type of molecule to another. That's the function of the enzyme. So those are four functions. Now the functions of the peripheral proteins. Peripheral proteins perform two very important functions. Those are important ones. There are other. Intercellular joining or junctions. I mentioned earlier that sometimes we see that the cells are attached to each other and the cell membranes of adjacent cells are attached to each other by some structures, protein structures. These are the intercellular junctions or joinings. So the cells are attached to each other. There are three types of intercellular junctions. Those are very common in our human body. So we'll talk about those three types of intercellular junctions or joinings. And another important function is cell-to-cell recognition. That is extremely important. Your body cells know if the cells are coming from your own body or from another person's body. You already know that, right? Like if I want to give my tissue to your body, most of the cases what will happen? Rejection, right? The chance of rejection is very high. Why? Because your cells know if the cells are coming from your own body or from outside another person's body. That is cell-to-cell recognition. Okay, and some peripheral proteins, those I showed you, just like this, they can recognize if another cell comes from outside. They can recognize if from own body or another person's body. That is the cell-to-cell recognition, and that is a big challenge, right? Like organ transplantation or tissue transplantation, right? Skin grafting. If I take skin from your body to graft to another area, it will be okay, right? But if I take from another person, then you have to take measures to stop rejection. So that is the cell-to-cell recognition. Very important function of the peripheral protein. Now, three types of intercellular joinings or junctions: tight junctions, desmosomes, and gap junctions. Tight junctions prevent fluids and most of the molecules from moving between the cells. So, that inside the cell you have the fluid and chemicals. So the tight junction doesn't allow the fluid or molecules easily pass through them. Only very small molecules and a small amount of fluid can pass through the tight junctions. So very tight. Desmosomes. Desmosomes are the junctions. They hold the cell membranes very strongly like this. You see here, this is the structure of the desmosome. Hold the cell membranes very strongly so they will not get easily separated. And gap junctions, its name is telling you, have many holes or pores. So through the gap junction, the ions or molecules or fluid can easily pass from one cell to another. So that's why they are called the gap junctions. Many pores or channels are present inside the gap junctions. So cells can quickly talk to each other through the gap junction. So those are three. One allows very little amount of fluid and very small molecules. That's the tight junction. Desmosomes hold the cell membranes very strongly together, and gap junctions allow the ions or molecules easily pass through them. So here you see how they look like. This one is showing the tight junction, structure of a tight junction. Just know that tight junction is, it looks like this. Two cell membranes, this is one cell membrane, this is another cell membrane, like two sheets of clothes. If you put them together and stitch them, you see there, that's the tight junction. And those tiny protein structures are forming the tight junction. They're like stitching the cell membranes together. Okay, so that's the tight junction. Those are called interlocking junctional proteins. Okay, desmosomes. Desmosomes are like the zipper of your cloth. Sometimes you use the zipper very strongly, put the pieces together, right? So if you see the structure of a desmosome, in the inner surface of the cell membrane, adjacent cell membrane, you have discs, and from the discs, the protein structures arise and get attached to each other. So like this. This is one disc, this is another disc, and the protein structures, those are called interlocking proteins or cadherins. Another name is cadherin. They arise from the disc and get attached to each other like this. Make sense? Holding the cell membranes very strongly like this. So you see here in the bottom two cell membranes, and in the inner surface the discs, the yellow color, and from the discs the blue colored, those are the linker glycoproteins or cadherins. So they get attached to each other very strongly. So where you will find the desmosomes? Example, in your heart muscle. In the heart muscle, we see plenty of desmosomes because you know when heart contracts, all muscle fibers should contract at the same time, right? Because heart pumps the blood. So when heart will contract, the muscle fibers must contract at the same time quickly. So they are very strongly held together. So all of them must, work together. Not like one will work at one time, another will work at a different time. That will not perform the function. So to contract the heart, you need all fibers working at the same time. So plenty of desmosomes we see in the heart muscle. In the skin, have you seen that if your skin gets very dry and you can feel the skin? In snake you will see when they change the skin, the whole chunk of skin, right, gets off. Why? Because those cells are attached to each other by desmosomes strongly, so will stay together. So that's the desmosome. Gap junctions. You see, through the gap junctions there are channels, so the ions or molecules can easily pass through the gap junction because of the presence of the channels. Gap junctions are also present in heart muscle. Why? Because again, heart muscles fibers must be attached to each other strongly by desmosome. Not only that, they need to talk to each other quickly. So the ions can easily be exchanged between the muscle fibers or cells. So those two types of junctions are heavily present in the cardiac muscle, desmosomes and gap junctions, and both are helping the heart muscle fibers to all of them to work together. Okay, so those are three types of intercellular joinings or junctions: tight junctions, desmosomes and gap junctions. Okay, now transport across the cell membrane. You all know that cell membrane is a semi-permeable membrane, right? Semi means partially permeable. That means the cell membrane allows certain chemicals to pass through it, but not everything. Selectively permeable. We say selectively permeable. Now, there are two processes of transport across the cell membrane. One is called the active transport, another type is the passive transport. Which one needs ATP? What do you think? Active, right? Active, its name is telling you that it needs energy. Active transport requires ATP, passive does not. Now, since active transport needs ATP, active transport can only occur through the living cell membrane. not in any other type of membrane. Non-living membrane will not, through non-living, no active transport will occur, because ATP is only produced in living body, right? Living cell, you know that. ATP is only produced by metabolism in living tissue, living cell. So, two things, active transport requires ATP, and active transport only occurs through the living cell membrane. But passive doesn't need ATP and can occur through any type of membrane, living or non-living. Now, active, let's first talk about the passive. Passive could be simple diffusion, facilitated diffusion, and osmosis, those three. So, what is simple diffusion? If you separate two solutions by a semi-permeable membrane, this is the membrane, semi-permeable membrane, and in this side you have a solution, concentration is high. In this side you have a solution, the concentration is low. Okay, so this is low concentration. Now, since this is high concentration, here the solute, concentration of solute is high, water is what? Less. Make sense? Because this is highly concentrated solution. In this side, you have water is what? More or less?
More.
More. But solute is less. Less solute, more water. In this side, more solute, less water. Is it clear? Okay. So, just remember one thing. Why the transport occurs in this case? To bring equilibrium, same concentration in both sides. That's the goal. If you just know that, you know other things. Now you tell me, if you want to bring equilibrium, same concentration in both sides, who should move from this side to this side? Solute, because solute is more here, right? So solute should move from this side to this side, from high concentration to low. Solute will move, and that is called simple diffusion. Now you tell me, water should move from which side to which side? This side to that side, right? Because water is more here. Water will move that way. So that is called what? Osmosis. So this is osmosis, the movement of water or solvent from low to high, and this is simple diffusion, the movement of solute from high to low. Make sense? So the goal is bringing equilibrium, same concentration. If you know that, it is easy who should move in which direction, right? So those two. Facilitated diffusion. You remember a few minutes ago I showed you that some integral proteins have channels, right? So those integral proteins are doing what? Facilitating the transport, right? So that's the example of facilitated diffusion. Some protein structures in the cell membrane, they help in the transport. Okay, so it is like, if you want to cross a lake or river, and if you know how to swim, okay, you just float your body on water, okay, you don't need to actively swim. Just, if the current is in your favor, then you will easily go to the other side, right? Even without spending energy, you can go. Now, if you want to swim against the current, then you have to do what? You have to actively swim, right? Utilize energy. So that is active transport, and without energy, utilizing energy, that's the passive. Now, sometime what you can do, you can use a boat, right, to cross. You can sit on a boat, and if you are going towards the current, then you don't need to start the engine. It will just go by itself, right? But you are not doing anything, you are sitting. So the boat is facilitating you, right, just to cross the river. Now if you want to come against, then you have to start the engine, ATP energy, right? So you need to use. So those are the transports across the cell membrane. This is an experiment that you can do, like take U-shaped test tube and put a semi-permeable membrane in the center, and in one side put high concentration solution, another side low concentration. Eventually you will see the concentration in both sides will be same, because the solutes will move from high to low, water will move from low to high, that I have shown you here. Okay, so that's one experiment. Here, this experiment is only for osmosis. Why? Because you see the semi-permeable membrane here, this membrane has very tiny holes or channels, smaller than the solute. So solute movement has been blocked here. No solute movement can occur because the membrane has the holes, those are smaller than the size of the solutes. So in this case, only water will move. That means only osmosis will occur, right? So you see that when only the water will move from low to high, that is the osmosis, and in one side the volume of solution will be more. The final goal is bringing equilibrium, and you see here the equilibrium has been established, although the volume is different. The volume is more here, less here, but concentration is same in both sides. Okay, so only water will move from low to high in this case, because you are not allowing the solutes to move. The response of a living cell in 3 different solutions. Now, if you put a living cell in a solution, this is the living cell, and this solution is hypotonic. Hypo means less or more? Less, right? Hypotonic. That means what? If the concentration, just for example, inside the cell is 10 milliosmol, this concentration is less than that. That's why it is called hypotonic compared to the cell. Make sense? So, for example, this one has 5 milliosmol. So inside the cell is 10, concentration is high, right? So this solution is hypotonic solution. Is it clear? So who will, or which way the water will move? Water always moves from low to high. That is osmosis, right? So water will get into the cell. Is it clear? From low to high. And the cell, the cell will get what? Bigger, enlarge, right? Swelling of cell will occur. So eventually it may burst, rupture, because it will get bigger, and can be ruptured. So that is the response of the cell in hypotonic solution. Water will get in and the cell will get enlarged. Now if you change the solution, now the concentration is 15. This solution is what? Hypo or hyper? Hyper. Now water will always follow the same rule from low to high. Make sense? Same rule. So in this case, what will happen? From low to high, water will get out, right? So the cell will shrink, will get smaller. Is it clear? So in hypertonic solution, water will get out from the cell and cell will get smaller or shrink. Now, if it is isotonic, that means same concentration inside and outside. So this solution is now called isotonic. In this case, no movement because equilibrium is already there, right? Equilibrium is there. So no movement of water will occur. The cell will not change the shape. So now if I ask you to put saline in a patient's body, into patient's body, blood, what type of solution you will prefer? If you give hypotonic, then what will happen?
Oh.
Iso, right? Yes, isotonic solution. Because otherwise the cell shape will change.